warm welcome to all of you present in the audience as we gather here today to celebrate our annual day function, marking another milestone in the journey of our institution. February 17th is a very important day because throughout the year we look forward for 17th February because it is important for both scientific feast as well as cultural feast. The day starts with Sir Edward Mellanby Memorial Oration, and as you know that this lecture series was started as a tribute to Sir Edward Mellanby, who was a British pharmacologist, and his research helped to establish the role of vitamin D deficiency in rickets. In 1951, he helped to establish CDRI and served as the first director of our institute. After the memorial, Edward Mellanby Memorial Oration, we have the annual day program, wherein we'll get the chance to listen to the chief guest of the day. And this day closes with a cultural evening wherein our students perform and give us the flavor of different cultural activities and give the flavor of our country. To begin the program, I would like to request our director, as well as our guest, Professor Sandhya Vishweshwaraya and Mr. G.V. Prasad, to come up on the stage. I also request Dr. Divya Singh, who is the convener of this program, to join us on the stage. On the behalf of CDRI family, I request Dr. Radha to please present the bouquet of flowers to our guest. Today we are also joined by lab directors of different institutes in Lucknow. Professor Prabodh Trivedi from Sister Lab, Director CMAP. I request Dr. Radha Rangarajan to please present the bouquet of flowers. And we are also joined by Director NIPER, Professor Shumbani Sharaf. Sister, please. Ma'am. I request ma'am to address the audience.
Distinguished guests, colleagues, friends, and my dear students, I welcome you all to our 74th annual day, and as well as to conduct the Sir Edward Mellanby Oration, as Neeti just mentioned. I extend a very special welcome to our chief guest, Mr. G.V. Prasad, co-chairman and managing director, Dr. Reddy's Laboratories, and to Professor Sandhya Vishweshwaraya, National Science Chair and Honorary Professor, Indian Institute of Science, who will deliver the Mellanby Oration. Thank you so much for accepting our invitation and taking time out of your schedule to be here with us. We're really honored by your presence. I welcome Dr. Prabodh Trivedi, Director CMAP, Dr. Shubhni Saraf, Director NIPER Raebareli, and other guests from Lucknow-based scientific and medical institutions. Thank you very much for joining us. I welcome our partners from MyLab Discovery Solutions, Dr. Makhan Kumar and Ms. Amrita Joglekar, and Professor Shona Chatterjee from Center for High Impact Neuroscience and Translational Applications, Kolkata, with whom we will sign agreements today. And of course, a very warm welcome to all of you, faculty, staff, and dear students. As you know, CDRI is a constituent laboratory of the Council of Scientific and Industrial Research, and was formally dedicated to the nation on 17th February 1951 by the then Prime Minister, Pandit Jawaharlal Nehru. CDRI's mission is to develop drugs, diagnostics, and process technologies, and to ultimately make healthcare affordable for people in India. Our faculty are engaged in basic and translational research, teaching and training, and offering research services to academia and industry. I thank you all for joining us today to celebrate our achievements of our 74 years. We will start today's program with the Sir Edward B. Mellanby oration. Today's oration, the 50th in the series, is being delivered by Professor Sandhya Vishweshwaraya, an acclaimed scientist who has made major contributions to our understanding of cyclic AMP and cyclic GMP-mediated signaling pathways in host pathogen interactions. Today, we will learn about the role of the receptor guanylyl cyclase, which converts GTP to cyclic GMP, in fluid and iron secretion in the gut, with implications in diseases such as diarrhea and inflammatory bowel disease. I'm sure you're all eager to listen to her talk. I will not keep you waiting. I now request Dr. Neeti Kumar to introduce the speaker and take the program forward. Thank you very much for being here.
It's an honor to introduce Professor Sandhya, who is not only a good person, a good teacher, and a very fantastic researcher. Her career was as following. She did her bachelor's from Osmania University, Hyderabad. Then she did master's in chemistry from IIT Kanpur in 1980s. Now this is the era where you would have handful of female researchers or female students in IIT Kanpur campus. After understanding the world of chemistry, she extrapolated that knowledge of chemistry to understand the chemistry of biological molecules, and she did PhD in biochemistry department from ISC Bangalore. And after her PhD, she worked for a few years in AstraZeneca. And then she shifted from industry to academia, and she joined molecular reproduction and developmental and genetics in ISC. She has various national and international recognitions, to name a few. She's an honorary visiting professor at Imperial College London. She's also an adjunct professor in University of Bergen, Norway. She's president-elect of IUBMB. As Dr. Radha mentioned already, she has served as National Science Chair, and she is a fellow of Indian National Science Academy, as well as a TWAS fellow. She's received fellowships from Human Frontier Science Program, and she is also a recipient of CDRI Excellence Award in 2005. And she has currently welcomed DBT Alliance fellowships. She really enjoys, I heard her YouTube videos, she really enjoys to interact with students and encourages public outreach to harness and channelize their talent. And she's a strong advocate of bridging the gender gap in STEM at all stages of professional career. So we look forward to listening to you, ma'am. And I request the dignitaries to please take their seat in the audience.
You know, it's really a great pleasure to be here. I last visited in 2005, which is almost 20 years ago, when you were still in the old building. and that was a great pleasurable moment for me as well. And I think I'd really like to thank all the staff, the director, of course, for the invitation, and all the other people who have been really making our stay so welcome here and so comfortable. Now, I think it's particularly fitting that perhaps I'm going to talk a little bit about gut function and gut physiology, especially given the fact that Sir Edward Mellanby involved himself so much in thinking about the diet and nutrition and its role in human health. Now, more than 2,000 years ago, it is said that Hippocrates first made the statement that all disease begins in the gut. Now, I think it's in the last couple of years that we've realized how true this statement is. And I think we're all aware of the fact that at any point of time, if we do have slight discomfort in our gastrointestinal system, we do feel rather uncomfortable, and it does affect our overall physiology and behavior. Of course, perhaps 1,000 years before that, Ayurveda has emphasized the importance of the gut in normal physiology, and a healthy gut, as described here, is important in maintaining our normal behavior and suits our dosha as one of the major things that Ayurveda talks about. I picked up this cover page of an article that Sir Edward Mellanby wrote many years ago about the relation of diet to health and disease. And this was a lecture that he delivered in 1930, almost 100 years ago. So I think, it's really important for us to realize what visionaries we had at that time who thought about health in a very holistic way and in terms of overall physiology and the importance of the gut. Now, as far as the gastrointestinal tract is concerned, we would all have drawn this picture perhaps at school onwards in our biology lessons. The gastrointestinal tract, interestingly, is one of the few orifices in our human body which takes in material which is non-sterile. Most of our body is relatively in a sterile environment, but it's the food that we take through our oral cavity that is non-sterile, and therefore the gut, as you'll come to know, handles that in a very, very sophisticated way. So the food passes from the oral cavity into the stomach and then into the small intestine, where a large amount of digestion as well as absorption of nutrients occurs. This then moves on into the large intestine or the colon, and finally any waste material is expelled, as we all know. When you think about the gut architecture, this is something that perhaps maybe even I appreciated as to how complex it is in terms of regulating function. We have the region of the gut that actually comes in contact with food and, of course, microbes as part of the microbiome. This is lined by a very sophisticated set of intestinal epithelial cells which prevent many of these harmful products from entering into the system. This is surrounded by a submucosal layer where there are a number of glands which produce various hormones which get into the gut. You have the muscular layer, and this muscular layer is controlled by a great degree of innervation by the nervous system. And it is this neuronal interface that is extremely important in bringing about what's called the gut-brain axis. And the neuronals, some of them, of course, control muscle movement, which allows the bolus of food to pass down the gut. And of course, there are a number of other ways by which the neuronal system affects brain function and brain output. Now, I'm going to focus mainly on the intestinal epithelial layer because that is where the system is that we're going to work with. Now, if you think about the small intestine, we all know that the small intestine is composed of crypts and villi, which increase the surface area presented so that a greater degree of food absorption can occur, as well as fluid and ion secretion. The intestinal epithelial cells are covered by a very thick mucus layer. Now, why is this mucus layer important? This prevents the arrival of some of the gut microbiome from impacting and touching directly the intestinal epithelial cells, because that will generate an innate immune response. And that innate immune response will then translate into overall inflammation occurring in the gut. So this mucus layer and the thickness of the mucus layer really determines how sterile the gut environment is kept. Of course, we have the microbiome, which is now becoming more and more important in terms of understanding of how our whole physiology is affected. We have a number of studies that relate to the characterization and identification of the gut microbiome. the various bacteria that are present, what the genes are in these bacteria that influence our behavior. The large intestine, or the colonic epithelium, is also covered by this very, very thick mucus layer. Again, the epithelial cell layer is composed of a variety of different cells, and I think this is very striking. When you think about how the epithelial layer is modified, right at the base of the crypts of both the small and the large intestine are a group of stem cells. Now, these are marked by a protein called LGR5, which is a standard stem cell marker. It is important for us to realize and think about the fact that the gut epithelium in our body is regenerated completely once in 4 days. So this is one of the most highly proliferative tissues in our body. And therefore, one must appreciate the fact of how well this replication is controlled in order to prevent tumorigenesis. As we're all aware, we have colorectal cancer and a number of tumors that do affect the gut. Most of us, of course, control this process very carefully. Adjacent to the stem cells are what they're called +4 stem cells. So these are cells that just are adjacent to at the lower level of the crypt. These are what are called transit amplifying cells. As these cells migrate up the gut wall, they start differentiating. And the differentiation occurs into many, many different cell types, which are extremely important for gut physiology. For example, some of these cells differentiate into secretory cells called goblet cells. Now, these are the cells that produce the thick layer of mucus. So any aberration or problem in the goblet cell will result in a reduction of the mucus layer and therefore possibly inflammation caused by the gut microbiomes present in the gut lumen. You have tough cells, which produce many hormones which are important for gut function. You have enteroendocrine cells also that are extremely important. And of course, a very large number of absorptive enterocytes which are involved in the uptake of fluid and ions. So as you can see, these cells migrate to the tip, and when they reach the tip, they undergo a process called anoikis or apoptosis, and that results in these cells floating off, releasing into the lumen, and they are replenished by a fresh lot of epithelial cells differentiating. Now, all of us in this audience, I'm sure, have experienced some form of diarrheal disease at some stage during our lives. Of course, we've always attributed this to something we've eaten outside or some contaminated food that we may have taken in. But it's very important to realize that this diarrheal disease is extremely severe and important in young children, especially below the age of 5, and in countries such as ours, low and middle income countries such as Asia, as well as sub-Saharan Africa. A couple of figures which might actually shock you: 1 in 10 child deaths around the world results from diarrheal disease just during the first 5 years of life. There are almost 1 million fatalities worldwide. Now, this is the fatality rate that has been going on for hundreds of years. So we all panicked when COVID came along and perhaps killed 5 million people over a couple of years. But we're talking about hundreds of millions of deaths caused by diarrheal disease over many, many years. There are 5 pathogens that have been acknowledged to be extremely important for causing diarrheal disease. One is rotavirus. This is a virus that infects usually very young children up to about first year of age. You have certain pathogenic strains of enterotoxigenic E. coli, or ETEX. Now, these strains of E. coli harbor a plasmid which encodes a gene which produces a stable toxin peptide, and that is going to be the main focus of this research. Certain species of Cryptosporidium, certain other species of E. coli called enteropathogenic E. coli. These are the 5 major pathogens that are important for causing diarrheal disease in children and in adults, indeed, worldwide. Now, very early on, it became apparent that many of these stable toxin peptides that I spoke about were the mechanism of action involved cyclic GMP. Now, cyclic GMP has always been considered the poor cousin of cyclic AMP. All of us would have been taught about G-protein coupled receptors during our graduation and post-graduation. All of us would have realized how they elevate cyclic AMP levels in cells. It was very interesting to know that when Sutherland first identified cyclic AMP, within a few years later, he identified cyclic GMP as also being an important second messenger. However, it's always had a slightly lower key in terms of understanding the signaling mechanisms that cyclic GMP regulates. These are the stable toxin peptides that I've talked about, which are produced by E. coli strains. Very interestingly, these are very small peptides, about 19 amino acids long, and they contain 6 cysteine residues, which are disulfide bonded. Now, it is this disulfide bonding that imparts the extraordinary stability to these peptides, and that is why they're called heat-stable enterotoxins. They cannot be destroyed by cooking the food, and so therefore that's exactly why they have become one of the major causes of diarrhea in countries like ours. Interestingly, for those of us who think about these things, these peptides are not produced as 19 amino acid peptides by E. coli. They're actually produced as pre-pro peptides. And this is a very, very rare occurrence for bacteria. I mean, usually bacteria are pretty businesslike. They produce peptides exactly as they need them. But for these peptides, they're produced as actually 72 amino acid long polypeptides, which then get their signal sequence cleaved. There's a pro region that's cleaved before these peptides are released into the lumen of the gut. It took almost 25 years before we realized what the receptor was for these particular bacterial toxins. And this is the receptor that I'll be spending quite some time talking about. This receptor is called receptor guanylyl cyclase C because it was the third member of this family of receptors that was identified by David Garbers in the 70s. Now, as a biochemist, it was the structure or the apparent composition of this protein that fascinated me when we first began the laboratory. These receptors are single-pass transmembrane domain spanning proteins, and they exist as preformed dimers in the cell. The extracellular ligand binding domain is the region where the heat-stable enterotoxin binds. These peptides are not cell permeable, and therefore you need a receptor with an extracellular domain for these peptides to bind to. The single transmembrane domain leads to a large intracellular domain. At this extreme C-terminus is the guanylyl cyclase domain. This is the region where GTP is converted to cyclic GMP. Upstream of that is a very long helical region, which we have studied quite extensively. We call that the linker region. And upstream of that is a region which has very significant similarity to protein kinases, and it's called the kinase homology domain. Why kinase homology domain? Because of the amino acid sequence similarity. However, there are critical residues which have been identified in protein kinases, which are important for the phosphotransfer activity. Now, those residues are absent or mutated in this region of the receptor. So while this receptor adopts possibly a conformation that is similar to protein kinases, it does not do any phosphotransfer activity. Essentially, ligands bind to the extracellular domain of this receptor and must induce some kind of conformational change, resulting in the increase of cyclic GMP levels in the intestinal epithelial cell. This is the highest resolution structure we have of this receptor. There is no crystal structure available of the full-length receptor. We have crystal structures of the extracellular domain, but they're in complex with antibodies, and so we think that this has pulled the receptor out of its regular conformation. There are structures of the intracellular domain, but interestingly, none of them delineate the guanylyl cyclase domain. So we are working here with a protein for which there is really no known detailed structural information. Obviously, this receptor was not present in the gut waiting for a bacterial enterotoxin to come along. It had to have normal gastrointestinal hormones that would regulate its activity. And these were peptides called guanylin and uroguanylin, which took another 20 years after the identification of the receptor to be able to characterize. These ligands have 1 log order lower affinity to the receptor than the heat-stable enterotoxins, and so you can understand that these bacterial toxins are superagonists of the receptor. Where is this receptor expressed? It's expressed in almost all cells of the intestinal epithelium, and therefore the gut is the major site for synthesis of this receptor or production of this receptor. However, we and others have identified that this receptor is also expressed in reproductive tissue as well as certain cells in the brain. Now let's get down to the signaling mechanisms which ultimately identify how these bacterial toxins cause diarrhea. Let's focus on what you can see as blue arrows here. Ligand binding to the extracellular domain of this receptor results in increased cyclic GMP production. Cyclic GMP now binds to a very specific protein kinase called cyclic GMP-dependent protein kinase 2. This is a kinase that is largely expressed in the intestinal epithelium. Activation of PKG2 following cyclic GMP binding now results in phosphorylation of two very important molecules involved in ion transport. One is CFTR, the cystic fibrosis transmembrane conductance regulator. This is a chloride channel. Now, phosphorylation of CFTR results in increased chloride efflux into the lumen of the gut. The second protein that cyclic GM or PKG2 phosphorylates is a sodium hydrogen exchanger. Now, phosphorylation of NHE3 inhibits NHE3, and therefore there is an accumulation of sodium ions in the lumen of the gut. Activation of CFTR results in increased chloride in the lumen. Inhibition of NHE3 results in increased sodium in the lumen. And this sodium chloride in the lumen of the gut creates an osmotic imbalance. And this osmotic imbalance now drives fluid secretion from the intestinal cell, and that is the cause of the diarrhea. Now, you've all have heard about how it's so important that when one has diarrhea, that one drinks plenty of fluid, also a pinch of salt and a little bit of sugar in these fluid therapies that are recommended. This is the explanation for that. Over the years, we and others have looked at other signaling events downstream of cyclic GMP and PKG2. One of the important things that we discovered was that there was a regulation of cell cycle progression by cyclic GMP. This was mediated by, again, phosphorylation of P38 MAP kinase, phosphorylation of the transcription factor SP1, which now regulates the transcription of P21, which, as we know, is a cell cycle regulator. So this whole mechanism is extremely important in preventing over-cell proliferation in the gut lumen. And in GCC knockout mice, that is mice which don't have GCC, you find an increased incidence of tumorigenesis, colorectal tumorigenesis, following stressors that are provided to these animals. Finally, very recently, the role of cyclic GMP in regulating neuronal function has been delineated by groups in the US, mainly Scott Waldman's group. So you see that there are many, many signaling outputs downstream of cyclic GMP mediated by this receptor. And most of this work has been delineated using GCC knockout mice, which I must mention are perfectly normal, except when you provide stressors. They could be carcinogens, they could be the stable toxin peptides. So it's important to realize that this receptor is important in maintaining normal physiology in the gut. Very interestingly, once it became apparent that activation of this receptor could result in increased fluid and ion secretion, certain drugs were developed called linaclotide and plecanatide. These are FDA-approved drugs, and these are nothing but the core peptide sequence of the stable toxin peptide from E. coli. So these drugs are used for the treatment of constipation, as you would expect, because this will allow this individual to elevate cyclic GMP levels in the gut epithelium, resulting in the signaling events that I just spoke about. Interestingly, these drugs are also utilized for the treatment of IBS, or irritable bowel syndrome, and they've even found effects of these drugs in pain or gut-mediated pain. So again, the neuronal connection comes in very closely here. Now, one of the things that intrigued us was this receptor is highly conserved, right from fish all the way to humans. But there was no known report of any human mutation that was associated with disease in this receptor. In 2012, I was approached by clinicians at the University of Bergen in Norway. They had identified a family with 32 members, where every individual in that family who was affected reported childhood constitutive diarrhea, and this diarrhea continued all the way to adulthood. Because they had good records, and of course they had samples from all of these individuals, they did classical genetic analysis to try to identify where the mutation lay in these people. Now, if you look at the tree, you can see that this is an autosomal dominant mutation, sex independent. When they did the analysis, I'll talk about that a little bit, but just to tell you how severe some of these symptoms were in these patients. Many of these patients reported 4 to 5 stools a day, some of them as many as 20. Now, this can be quite inconvenient, as you can imagine. Many of them had to undergo laparotomy in the gut because there was severe inflammation and problems in the ileal region, which resulted in ileal inflammation, ileal resection. There were anatomical variants at the ileal-cecal junction, and many of them were diagnosed with Crohn's disease, which many of you would have heard of, which is an inflammatory disease in the bowel. So when they did the genetics, they realized that the mutation lay in a region of chromosome 12. And when they looked at that particular region of chromosome 12, the only gene that they could identify which was associated with diarrhea was receptor guanylyl cyclase C, which in humans is GUCY2C or GUCY2C. They then decided, okay, this is the only gene that's there in this locus associated with diarrhea, so they sequenced the whole gene. And they found that in affected individuals, there was a single point mutation which converted a serine residue to an isoleucine in the guanylyl cyclase or the catalytic domain of this receptor. At this point, they wrote to me, because by then we had been publishing a lot about mutational analysis of the receptor, so they wrote asking whether we could characterize what the effect of this mutation did on receptor function. And as you might expect, this mutation, a single change in residue, resulted in hyperactivation of the receptor. So you can measure cyclic GMP production in wild type and mutant cells, and you can see that when the cells expressing the mutant receptor, levels of cyclic GMP produced were much higher, which would have resulted in the diarrheal disease or increased activation of the signaling pathway, and therefore the diarrhea associated with these patients. Now, this was the very first report of a disease associated with receptor guanylyl cyclase C. Now, one of the advantages of publishing in journals like the New England Journal of Medicine is that a number of clinicians read such papers. And therefore, a couple of years later, I was approached by clinicians at the University of Innsbruck in Austria. They had identified 4 children with constitutive childhood diarrhea. And this is the kind of picture that they saw in the child as soon as it was born. This bloating of the peritoneal cavity is because the fluid from the child's gut cannot escape anywhere else except into the amniotic sac, or the peritoneum. And so there's a huge amount of fluid accumulation, which could be seen even in the mother during her regular sonography. What that meant was that as soon as a child was born, the fluid from the peritoneum had to be drained because the child was unable to breathe. There was so much pressure put on the lungs. They identified that based on our publication, they realized that this was a mutation that possibly could be in GCC. They sequenced the GCC gene, and they found another mutant, which was an arginine to a serine mutation in the linker region, which I spoke about in the receptor. Now, 20 years prior to their publication, we had made systematic mutations in the linker region of this receptor because we predicted it to be alpha helical in structure, and so we decided that we would mutate that to proline, all the residues to proline, purely to break the helix and look at the consequences of those mutations on receptor function. And we found that mutation in the linker region, in many cases, led to hyperactivity of the receptor. This residue, R792S, lies in the linker region. They wrote to me, we made the mutation, and this was a very extraordinary mutation. Note here that even in the absence of ligand, basal levels of cyclic GMP were very, very high. So this was a constitutively active mutant. Further, in the presence of the ligand, and we use the stable toxin for our work here, this resulted in even super elevated levels of cyclic GMP and perhaps overactivation, or definitely overactivation of the signaling pathway. One point to note here is that these individuals, they were not infected by the stable toxin peptide. This misregulation was happening because of the endogenous ligands, guanylin and uroguanylin, produced in the gut. So these ligands are constitutively produced, activate the receptor to maintain regular and required fluid and ion secretion. When the receptor is hyperactivated, you have the diarrhea. Of course, subsequently, we received a number of other mutations that information from various clinicians around the world. And this is another mutation in the linker region at glutamine to 769 proline mutation. And the minute the parents wrote to me about this mutation, we knew exactly what it had done because we had made this mutation in our earlier work. So this really sent a chill up my spine that, you know, as pure biochemists and interested in looking at protein structure, we had accidentally made a mutation, which later on was found to be extremely important in the cause of human disease. Of course, over the years, a number of other variants have been associated with disease in humans in this receptor. Meconium ileus is the inability of a child, a newborn infant, to pass the first stool. And therefore, this stool needs to be removed surgically. This meant that there was constipation in that newborn child. And many of these mutations in GCC associated with meconium ileus, as you might predict, are inactivating mutations. So therefore, you have a whole set of mutations. Very interestingly, please note the number of mutations that were identified in the kinase homology domain of the receptor. As I said, this receptor does not do any phosphotransfer activity, but it appears to be an extremely important allosteric regulator, not just of the cyclase domain, but interestingly also of the extracellular domain. And we have characterized many of these mutations over the years. They lie all along the receptor. This is a structure predicted from alpha-2 by my collaborator at the University of Bordeaux, who's made it his mission to try to develop or understand the structure of this receptor, and I hope we see some progress in the next few months. Now, as a result of all of this work, mutations in GUSI2C have been recognized as a contributing factor to early onset inflammatory bowel disease. And that is because many of these pediatric cases that report to clinicians are also associated with severe inflammation in the gut. What I'm showing you here is a study from the Hospital of Sick Kids in Toronto. They analyzed around 1,000 pediatric IBD patients that they had obtained, did whole exome sequencing to identify mutations in various genes associated with IBD, and you can see that amongst 6% of the identified known variants were associated with GUSI2C. Of the 1,000 patients, I must emphasize that only 14% showed known variants in genes. What this means is that there is still a very large amount of information that is really not clear as to the major causes of inflammatory bowel disease. Because early onset IBD is likely to be a result of genetic factors rather than the environment, because adult inflammatory bowel disease could be a consequence of drinking, smoking, all sorts of other stuff that happens as we grow older, we feel that looking at the genetics of pediatric IBD is likely to allow us to identify new variants in future. Now, very interestingly, IBD and Crohn's disease was thought to be a disease associated with the Caucasian population because it was largely diet regulated. However, the incidences of this are increasing even in countries like India, where we do actually have enough in our diets, if we eat a normal diet, which protects us a lot from these disease. As I mentioned, there's a lot of inflammation that's associated with this particular receptor mutation. How is inflammation occurring? I talked to you only of the signaling pathways that appeared in the intraepithelial cells. And so, therefore, we decided that we should generate mouse models which could look into more detail at the molecular mechanisms that could regulate inflammation in the gut. We generated a knock-in mouse model, and this is a mouse model that harbored the Norwegian mutation, which is at a position serine S840 in humans. It's S839I in mice because there is one residue missing in the linker region. There's otherwise extensive sequence similarity between mouse and human. One thing I'd like to emphasize to all of you, especially in this audience, is that when we started looking very closely at the mouse receptor, it differed in biochemical properties quite significantly to the human receptor. There was a log order lower affinity of the receptor to the peptide. There was reduced guanyl cyclase activity of the mouse receptor than the human receptor. And these mice that we generated don't get diarrhea the way the humans do. Mice have an inbuilt ability to regulate the amount of fluid secretion in the gut. Why is that? Because their diet is different to humans. And evolutionarily, there have been adaptations in possibly many molecules in the gut which allow rodents to retain fluid in the gut. And one of those mutations could have happened in GCC because guanylin and uroguanylin are conserved in the mouse. But by reducing the affinity of these peptides to the receptor, there is a reduced level of activation and therefore reduced fluid secretion into the mouse gut. So mice are not humans, as we all know. So we characterized this mouse model. I'm not going to spend a lot of time on some of the details, but we first began, of course, by looking at intracellular cyclic GMP levels in epithelial cells produced from this mouse. And as you'd expect, the levels in the mutant mice were higher than in wild-type mice, both in the ileum as well as the colon epithelial cells. Now, as I said, mice don't get diarrhea, but there are ways that humans can actually measure whether these mice have abnormal gut function. You can send your students to the animal house and ask them to collect the feces that these mice produce in around 10 minutes. So you take these mice, put them in a clean cage, and over a period of 10 minutes, count the number of fecal pellets that are produced. And you can see that in our mouse model, the number of fecal pellets were produced were more. This could reflect the diarrhea that is seen in humans, but it's important to remember that this could also reflect aberrant neuronal function in terms of pumping out pellets at a faster rate. We looked at the water content in these fecal pellets. So you can take these pellets, lyophilize them, and weigh the pellets before and after lyophilization. And you can then calculate the amount of water in these fecal pellets, and you can see that that was increased as we would predict and hoped for in these animals. Finally, you can look at what's called total transit time. This is the time it takes for anything introduced orally to come out in the feces. So what you do is you gavage these mice with a red dye and patiently sit there and wait for the first red fecal pellet to emerge from these mice. And you can see that in the case of mutant mice, the time taken to remove the first red pellet was reduced. Again, the kind of diarrheal phenotypes that you might expect. But when you look at the overall more molecular changes, we looked, of course, at the fecal microbiome. Now, you can't do gut biology or gut physiology these days without looking at the microbiome. So we looked at the microbiome. And what you can see here is that many of the good bacteria, such as lactobacillus, colobacter, all of these were reduced in our mutant mouse. And bad bacteria, now by bad I mean in inverted commas, are bugs that are associated with Crohn's disease and inflammatory bowel disease in humans. And you can see that they all showed a significant increase. Now, you can dig deeper into the microbiome, which we haven't done so, but this was sufficient to report that changes in the gut lumen resulted in either the proliferation or the reduction of certain bacteria, which could also impact in the overall disease. When you look at additional molecular changes, which came out of RNA-seq analysis, you can find a number of genes that are misregulated. Many of these genes are inflammatory-associated genes, and you can correlate some of the gene expression changes as those seen in mice in which colitis has been introduced. For example, you can feed mice with DSS, which induces colitis in animals, and therefore you can see that some of these gene expression changes were similar. And importantly, many of the gene expression changes were those that had been reported in human patients with Crohn's disease and inflammatory bowel disease. So therefore, we had a mouse model, which was a first preclinical model for understanding inflammatory bowel disease mediated by this receptor. And we reported on dysbiosis, which very interestingly could be a consequence of the altered and fluid ion secretion in the gut lumen. Now, what happens in the small intestine? I told you that the small intestine was a region which was severely affected in these animals. You could look at epithelial barrier permeability, and we could see that the epithelial barrier permeability was increased in these animals. What this would have meant is that the microbes or the microbiome would now have greater access to the epithelial cell and therefore induce an inflammatory response. Paneth cells are extremely important cells which produce defensins and antimicrobial peptides which control the microbiome composition. And in our mice, we found a reduction in Paneth cell function. Now, Paneth cells can be looked at in immunohistochemical staining of the gut by staining for lysozyme, which is a major protein produced by Paneth cells. And you can see that lysozyme production was significantly reduced in the mutant mice. Moreover, many of the defensins, which are important antimicrobial peptides, also showed a reduction in their gene expression in the Paneth cells. So there was compromised Paneth cells in these animals. And of course, correlated with all of these changes, we could measure pro-inflammatory cytokines by ELISA in gut extracts prepared from these animals, and you can see that many of the pro-inflammatory cytokines, such as interferon gamma, TNF-alpha, they were all significantly increased in the lumen of these mice. So we now had an animal model system where we could also look at inflammation brought about by mutations in GCC. How can we now become even more cell-intrinsic? Can we start looking at changes that happen in the gut epithelial cells as such? And so therefore, we took recourse to generating gut organoids. Now this, of course, is work that's been done and established by Hans Clevers and his group in the Netherlands for many years. We prepared gut organoids. Now essentially what you do is isolate crypts from the small intestine of these animals. These can be cultured in vitro in the presence of very critical growth factors, and then you actually develop what are called mini-guts. Now, I'd like you to note one important thing, that these mini-guts could be considered inverted guts. And the reason is that the lumen is now within these organoids, and the epithelial cells are actually containing that luminal region. When we looked at the mutant receptor in these animals, we found that there was a leftward shift in the ability of this receptor to respond to the ligands ST as well as uroguanilin. So the mutant receptor, in some way, in both humans and in mice, was more sensitive to the presence of the receptor. Now, we're likely to understand this once we see the structure, but this becomes important when we start thinking about these organoids. Now, these are organoids prepared from wild-type mice. I hope the video works. No? Oh, dear. Yeah, I'm sorry. I think I'm going to get out of the screen show so that I hope you can see these videos. I don't know why they're not. Yeah, I'll run them here. This is a wild-type organoid treated with uroguanilin. Can you see the swelling and the ballooning of this organoid? Now, this is happening because fluid is being secreted into the lumen of these organoids. This is the fluid accumulation that is seen in the mutant organoids at concentrations of about 10 to the power of minus 5 molar uroguanilin. Now, when you decrease the concentration of uroguanilin, wild-type organoids over the time of time-lapse video imaging, which was around 14, 15 hours, you can see that there is very little accumulation of fluid in the organoids prepared from wild-type mice. But when you look at the mutant organoids, you can see that even at very lower concentrations of uroguanilin, the swelling is still happening. And this is exactly the reason why you see this diarrhea or apparent diarrhea in these models. And therefore, we now have organoids as a good model to study. And I must mention that we've started looking at the overall gene expression in these organoids by RNA-seq analysis. Is there any possibility of a therapeutic for treating these kinds of disorders? We were intrigued by very early data that came out, of course, from work in India at that time, that zinc is a very good therapeutic, cheap therapeutic for the treatment of childhood diarrheas. And WHO has in fact recommended the application or supplementation of zinc for treatment of childhood pediatric diarrheas. And there are various reports over the years. Zinc, of course, being an essential trace metal, has a number of effects in the cell. And dietary zinc has been shown to improve intestinal barrier function, which I told you was compromised in our mutant mice. And patients with IBD normally show zinc deficiency. So we asked ourselves, if we treat our mice with zinc, is anything going to happen? So we provided zinc in drinking water at concentrations which are known to be used for human therapy. And lo and behold, things started working. We could see that levels of intracellular cyclic GMP were reduced following zinc ingestion by these mice. The bowel frequencies were reduced in these mice, and fecal water content was also reduced in these mice. So therefore, this is really an alternative for treatment of some of these children. We're communicating with clinicians about these findings. But how does zinc work? how is zinc doing this in a GCC-mediated disease? And what we discovered is that zinc could now inhibit the activity of guanylyl cyclase, and this is because guanylyl cyclases are metal-dependent enzymes with magnesium at the active site, and what we think is happening is zinc is replacing magnesium and not allowing the catalytic conversion of GTP to magnesium, to cyclic GMP. What was even more interesting for us is the fact that zinc affinity for the mutant receptor was almost 1 to 2 log orders higher. So again, this mutation was doing something to make zinc bind better and bring about more effective inhibition. We could see this inhibition in the human receptor as well. Now, very interestingly, almost 20 or 30 years ago, we had looked at the effect of zinc on the catalytic activity of the receptor, and we published that in a very obscure, not obscure, it's a good journal, Biochemistry, but it was data that we kind of forgot about until we started developing these mouse models. So the message for many of the students, and of course the faculty in the audience, is what you do today may actually have implications 20, 25, 30 years later. So don't give up in thinking about how important your work is. What about organoids? Well, we put zinc on our organoids, and you can see that while you see swelling in the presence of mutant organoids, even in the presence of 1 micromolar uroguanylin, in the presence of zinc, that swelling was inhibited. And therefore, we think that this method might really work. So where are we now? This is just, I'm winding up now. Where are we now for the treatment of GCC-mediated disease? You can obviously think about various drugs that could be used or developed. Again, making sure that the pediatric IBD is a consequence of cyclic GMP production in the gut. As I said, there are many genetic causes for pediatric IBD, but if you identify mutations in receptor guanylyl cyclase C, it becomes important to study at what level that activation is happening before you provide personalized therapy. Of course, whole exome sequencing is something that we are doing right now with the pediatric population in India. We hope to be able to identify variants, if not in GCC, in any genes that may be associated with Indian pediatric IBD. And of course, once those mutations are identified, we can proceed to do in vitro characterization as well. Therefore, we have a lot more further molecular analysis to do in mouse models. We would like to look at the susceptibility of these mice to further infection using other pathogens. And the reason is that it's known that children with infantile diarrhea mediated by stable toxin enterotoxigenic E. coli are also more susceptible to infection by enteropathogenic E. coli. So what is the means by which that infection is regulated through cyclic GMP is something that we'd look at. And of course, the gut is the largest immune organ in the body. There are a whole host of immune cells that are found in the submucosa and in the mucosal level of the gut. And of course, we need to look at some of those in terms of thinking about inflammation at the gut level. As I said, we'll be doing whole exome sequencing of pediatric IBD patients in individuals, and we'd also like to be able to generate human organoids from some of these patients to be able to test in vitro therapies in the lab. As I mentioned, the structure of GCC is something that we're very, very keen to see. Even if we don't get it, I'd love to see the structure from another group. My collaborator, Pierre Maisonneuve at the University of Bordeaux, is working very hard on trying to determine structures. And of course, because we have the gut organoid model, this is amenable to high throughput screening using a variety of drugs. Finally, none of this work would have been possible without the really exceptional PhD students and postdoctoral fellows that I've had in the lab. Names underlined are those who graduated from my laboratory with PhD. You might recognize some of their names. Rachna Bhandari, who's in CDFD, was one of the first to start working on the role of the kinase homology domain in this receptor. I've received funding, of course, in the early years from the Government of India. I acknowledge the Department of Biotechnology, the Department of Science and Technology, and of course CSIR, because CSIR grants were guaranteed in the first years of a new laboratory setting up. And while the funds at that time were meager by comparison, they came on time. In the April, you received funds. And that, I think, was a huge boost for some of us as we were setting up our lab. So thank you, CSIR, for doing that. Subsequently, of course, based on the kind of work that we've been doing, we've been able to attract some international funding. The Bill and Melinda Gates funded us for generating a new mouse model to mimic the diarrheal phenotype. Not the mouse model I spoke of, that was funded by the Norwegians, but the mice are in my lab, but other mouse models. I was fortunate to get funding from the India Alliance DBT Wellcome Trust as a Margdarshi Fellowship, which allowed me to foster interactions between my lab and clinicians in Christian Medical College, Vellore. And of course, the Norwegians were very excited about all of this work, and so we received funding from the Research Council of Norway to develop vaccines for stable toxin-mediated diarrheas. Subsequently, I've had funding from the Royal Society as a collaborative grant for research professors, where I work with Professor Gad Frankel at Imperial College. And very recently, I've obtained an NIH R01 grant looking at a little bit more in detail at the molecular regulation of intestinal function by cyclic GMP. And for that, I collaborate with a former PhD student of mine, Dr. Avinash Shenoy, who's a reader at Imperial College right now. And of course, Pierre, who I mentioned at the University of Bordeaux. Let me close and thank you all very much for this kind invitation. It's been a privilege to have delivered this lecture. That's it. Should I take my... I think we can take, yeah. I don't want to pull anything out and mess up the... Thank you so much. Stay here. Okay.
Thank you, Dr. Sandhya, for taking us through the wonderful world of cyclic nucleotides and how they affect the gut health. It's really nice to see how simple biochemistry work can get connected to translational research, and the audience, I'm sure, is going to appreciate the entire journey and how that connect is eventually made after 20 years of work. Yeah. So with this, I would like to request the audience to give a big applause to Dr. Sandhya. And now I request our director, Dr. Radha Rangarajan, to felicitate the speaker. And I also request Dr. Prabodh Trivedi and Dr. Sharaf to please join Dr. Radha Rangarajan. And Mr. G.V. Prasad, they can all come on the stage. So now we'll break for... Okay. So we will break for tea and assemble at 11:25 for the annual day program. Manu, we'll give you a bag.
Thank you. Thank you.
Connect my computer. I got my machine.
If you're comfortable. So welcome back. We will now proceed towards the 74th Annual Day celebration. And for that, I would like to request Dr. Radha Rangarajan to come up on the stage and present the annual report.
Thank you very much, Neeti. Distinguished guests, faculty, staff, students, welcome back to the second half of today's program. After a breathtaking first half, and I have to say breathtaking because the span of work that Professor Vishweshwaraya presented, the multiple connections between basic research and clinical research, leading to a story that has so many implications in how we deal with IBD and childhood diarrhea. That was incredible, and it was masterful storytelling. Thank you very much. I also want to thank Dr. Saman Habib for bringing Professor Vishweshwaraya to CDRI for this event. So it is now my honor and privilege to present to you a progress report of the Institute. I can only present to you some highlights because we have between 20 and 25 minutes, and I might overshoot. These are not to make anyone feel that they are left out, but to give you a flavor for what's going on in the Institute. As you know, CDRI has both translational and basic research capabilities, and our faculty are involved in understanding disease pathogenesis and in identifying solutions, as much as in identifying solutions to treat and reverse the onslaught of disease. So we have both things. This makes us a truly unique organization. It also means that we're on a road that is less traveled. We must navigate the road to the best of our abilities, learning from the past, looking to the future, making sure that the road leads us to some meaningful destinations. As Robert Frost once said, "Two roads diverged in a wood, and I took the one less traveled by, and that has made all the difference." As we look back on our 74 years, we can say that we have made a difference. 13 drugs developed, 80 plus process technologies transferred to industry, collaborations with marquee partners from the pharmaceutical industry such as Dr. Reddy's Laboratories, Cipla, Zydus, Themis, and many others, a rich publication and patent portfolio, more than 2,000 students who have completed their PhDs here. Having said that, let's look at what we did in the last year. I'll start with some licensing and tech transfer updates. I'm happy to share that we signed 2 agreements in the last year. The first is for a phytopharmaceutical drug for non-alcoholic fatty liver disease derived from Picoriza crua, a plant that is known for its hepatoprotective properties. This has been developed in collaboration with CSIR-IHBT and is currently being evaluated in multicentric phase 3 clinical trials. If the trial meets its primary endpoints, Themis Medicare will seek marketing authorization and undertake the manufacture and sale of the phytopharmaceutical drug. Congratulations to the team led by Dr. Vivek Bhosle on the clinical side and Dr. Kumarvelu on the preclinical side. Our second licensing agreement was with MyLab Discovery Solutions. Our team, together with a team from KGMU, has developed a novel set of primers and probes to enable the detection of Zika and chikungunya, together with MyLab's primers and probes for the dengue virus. The kit addresses the need to identify pathogens for diseases with overlapping symptoms with one test. Congratulations to the team of Dr. Atul Goyal, Dr. Ashish Arora, and Dr. Neeti Kumar, as well as Dr. Amita Jain from KGMU. I want to thank the business development team led by Dr. Naseem Siddiqui for his able leadership and for enabling these deals. Moving on to ongoing translational projects, I will highlight 4 advanced lead candidates from our current portfolio. S0111793. This is an antimalarial compound for which non-rodent toxicity studies will commence shortly. This, as you know, is the last step before an IND filing. We are generously funded by BFI Biome and supported by Medicines for Malaria Venture. Congratulations to the team led by Dr. Saman Habib for overcoming many challenges to bring this compound to this stage. S0161348. This compound is being targeted as therapy for colorectal cancer. The compound promotes apoptosis by mimicking a protein that performs the same function in the cell. We are generously supported by the Department of Health Research for regulatory studies to support an IND filing. We also gratefully acknowledge the help and support of Origin Oncology. Congratulations to the team led by Dr. Deepak Datta and Dr. Deepankar Kole for their exceptional efforts on this project. CDRI 4-105, this compound is in the late lead optimization stage. It is a first-in-class compound for the treatment of neuropathic pain. Originally obtained from a screen of compounds from marine sources, extensive optimization was conducted to arrive at this compound. We have now conducted extensive efficacy studies and mechanistic studies. And we know this compound disrupts a receptor in the pain signaling pathway. Congratulations to the team led by Dr. Prem N. Yadav and Dr. Sanjay Patra for their outstanding work. S0190277, this compound is in the early lead optimization phase. It is exceptional because it addresses an unmet need that has been largely forgotten by the world, but which lurks in our country, filariasis. India has 40% of the global disease burden. We've identified a compound that is novel, has antifilarial effects in vitro and in vivo, and its mechanism of action is being worked out. Congratulations to the team led by Dr. Mrigang Srivastava and Dr. K.V. Sashidhara for their perseverance in working on a compound for a very complex indication. Thank you to all the team members, particularly the students. Your persistence, dedication, and hard work have played a major role in the advancement of these compounds, and we are grateful to your efforts. I also want to acknowledge the Translational Research Group, which is the internal committee that reviews emerging data and facilitates the progression of compounds in the portfolio. Additionally, I want to acknowledge and thank the in-house projects committee that helps with the review of proposals for in-house funding. This scheme allows for novel and innovative ideas to be tested for initial proof of concept before they become suitable for external funding. Thank you. I will now switch to basic research highlights. Novel drugs addressing unmet clinical needs come from high-quality basic research. I'm delighted to share that in the last year, we ran 107 basic research projects. Our faculty received funding from all the major funding agencies such as ANRF, DBT, ICMR, DST, Ministry of Earth Sciences, and DRDO, as well as private agencies such as BMGF, Bill and Melinda Gates Foundation, and BFI Biome. A total of 266 publications were published with an average impact factor of 4.9. Of these, 4 publications were in journals with an impact factor above 13. One of our publications also got selected for the journal cover. In addition to publications, the Institute filed 10 patents in India and 15 abroad and was granted 4 Indian patents. Congratulations to all the faculty. My thanks to Dr. Ajay Srivastava, IP Coordinator, for diligently supporting all the patent-related activities. I will now talk about CSIR missions and major grants that we've received. Mission programs are multi-institutional programs within CSIR, which are focused on common goals for all the participating institutes. We are currently contributing to 8 CSIR mission programs and leading 3. The first is the Pan-Cancer Mission, focused on identifying new anti-cancer drugs and enabling affordable therapies through process innovation for triple-negative breast cancer and ovarian cancer. The mission is now completing its 4th year with impressive outcomes. 1 candidate compound in regulatory studies, 2 processes developed, one for olaparib and one for palbociclib, multiple lead compounds, 12 patents filed, and close to 20 publications. We are also leading a multi-institutional effort to understand neuroinflammatory processes. The goal is to validate mechanisms regulating pain, neuronal regeneration, senescence, and degenerative diseases. The third mission project is in the area of antimicrobial resistance, where the focus is on identifying novel lead compounds, elucidating mechanisms of action for repurposed compounds, and establishing new methods for surveillance beyond hospitals. In addition, we are involved in other multi-institutional initiatives with funding from non-CSIR sources. These include a Center for Marine Therapeutics from the Department of Pharmaceuticals, a Center of Advanced Research for AMR from ICMR, a Center of Excellence in AMR from DST, and a drug discovery project under the Deep Ocean Mission of the Ministry of Earth Sciences. We have a project to validate non-hormonal targets for contraception funded by the Bill and Melinda Gates Foundation, and a set of early-stage discovery projects across therapeutic areas funded by BFI Biome. All put together, we received 48.5 crores from CSIR for direct research costs, rupees 21.58 crores from non-CSIR government sources, 9.8 crores from foreign agencies, and 1.38 crores from private Indian agencies and industry. We are very grateful to all our donors for their support. I want to thank the project management team led by Dr. Anand Kulkarni for managing a large number of projects and ensuring our adherence to the complex rules and regulations of each funding agency. We are also grateful to our Research Council headed by Dr. Bal Ganesh, the Management Council, and the Lab Strategic Group for their valuable support and guidance for our programs. I will now talk about some individual and institutional awards. Several individual awards were won in the past year. We are very proud of our faculty and our students who have been recognized. I will not be able to read out every one of the names because there are so many. We are proud of you and our hearty congratulations to all of you. Please accept our best wishes. However, there are two awards that we are particularly proud of that I do want to talk about. ICMR recognized CDRI for research excellence in the Best Extramural Institute category. The award recognizes the overall impact of the work of CDRI scientists in the biomedical arena. A second award that is noteworthy is from the Confederation of Indian Industry, CII. We were recognized as an institution of excellence for women in STEM. These awards recognize our collective efforts. Congratulations to everyone. I will now report on the status of the workforce and student affairs. During the year, a total of 9 scientists, 5 technical officers, 3 administrative staff, and 79 research scholars joined our institute. I cordially welcome all of them. During the year, a total of 29 staff superannuated. We acknowledge their contributions and offer our gratitude for their service. 91 research scholars completed their PhD work. Congratulations to them and their supervisors for achieving this milestone. I thank the Academic Affairs Unit led by Dr. Ritu Trivedi for driving several student-friendly initiatives in the past year and facilitating smooth passage for the students. Under the Skill Development Program, a total of 303 students, 66% of them women, received training at CSIR-CDRI. I'm grateful to the Skill Development team for managing these programs with great dedication. Our institutional outreach activities such as Jigyasa, Health Awareness, and Vigyan Jyoti programs have allowed us to connect to more than 5,000 students. I'm grateful to Dr. Sanjeev Yadav and his team for their continuous efforts. I will now report on key operational highlights. CDRI runs smoothly due to the relentless efforts of a very large number of committees. These committees deliberate and recommend decisions on issues ranging from purchase, infrastructure, security, selection for posts, contracts, and so on. Thank you to all the committees for your sustained and unwavering efforts to ensure that CDRI runs efficiently every single day. In particular, I want to acknowledge the extraordinary efforts of the Animal House Committee, headed by Dr. Amir Nazir together with Dr. Rajdeep Guha, for undertaking a major upgrade of the laboratory animal facility and creating an excellent training program. Thank you. We have renovated our canteen, which will be inaugurated by the Director General on February 19th. We are grateful to the Canteen Committee, headed by Dr. Rajkamal Tripathi, for transforming the look and feel of the canteen. Thank you. The Dispensary Committee has helped streamline medical services to the CDRI community and retirees of all Lucknow labs. We are grateful for their efforts. We have a new website, which we hope makes navigation easier. Thank you to the website committee headed by engineer Kural Srivastava for bringing this effort to fruition. I'm thankful to all the heads of the divisions and area coordinators for their contributions to the Institute. I acknowledge also the collective efforts of the Senior Controller of Administration, Controller of Financial Administration, Controller of Stores and Purchase, Head IT, Head Civil and Electrical Divisions, and their dedicated teams. Thank you very much. I also want to thank the team that helped with all the photographs and so on for this presentation today. Thank you. And last but not the least, I want to thank the organizing committee for today's program, headed by Dr. Divya Singh and Dr. Mrigang Srivastava, for their careful planning and meticulous execution, and perhaps some sleepless nights, but all is going well. Relax. In the last part of my talk, I'd like to pause for some reflections and talk about the future. We are entering our 75th year, so it is apt to ask, where do we want to be in 25 years? What capabilities do we need to acquire to get us there? And who will our partners be? Let me share some perspectives. If we look back, we can perhaps see some distinct phases in CDRI's evolution. The first 25 years could be summed up as the early years, when the institute was getting set up. The focus was on building infrastructure, recruiting scientists, setting up functioning laboratories. There was a lot of work done on natural products. The second phase, starting perhaps somewhere in the 70s, was focused on developing processes for bulk manufacturing and developing drugs for a nation that was grappling with a high burden of infectious diseases and high fertility rates. More than 80 process technologies were transferred to industry and 10 drugs went to market. The two best-known drugs from that period are Cencroman, which is a non-steroidal oral contraceptive still in the market with about 600,000 users, and Artether, which is an anti-malarial drug and sold currently in over 20 countries. Well, this phase ended, and a third phase began, perhaps in the late 90s or early 2000s. I'm piecing this together from reading annual reports of the last several decades. This phase focused on building basic research capabilities. This became necessary as drug discovery frameworks were changing globally. In his book, Emperor of All Maladies, Siddharth Mukherjee traces the history of treatments for cancer. Starting in the 1950s, drug discovery went from being a biology-blind, chemistry-driven approach to one that became driven by biology, culminating in the first targeted drug, imatinib, for chronic myelogenous leukemia. Caused by a swapping of segments of chromosome 9 and 22, leading to a constitutively active BCR-ABL kinase, imatinib was found to be a relatively selective inhibitor and demonstrated to have remarkable results in humans. Such successes heralded a new era in drug discovery. This, together with other factors such as the availability of funding and changing regulatory processes, led to a shift in focus at CDRI towards more fundamental research. Now, as we begin the march to the next 25 years, we must actively chart a course of balancing basic research and drug development. The former DG of CSIR, Dr. Mashelkar, often said, "Innovate, don't imitate." In fact, we are now well positioned to run our twin engines of basic and translational research to innovate and deliver more consistently. I dare say we can file 10 INDs in the next 25 years, and if we do our work well, stay focused on the unmet need, and ensure differentiation, we should be able to take at least half of those into phase 2 and beyond. The second question we must ask is, what do we need to get there? Undoubtedly, our approach must embrace technology wherever possible. Drug design, peptide design, prediction of protein structures, computational screens, structure-guided drug design. We must take advantage of every possible technology, including AI, to reduce the time and cost of discovering drugs. We have ongoing collaborations with IIIT Delhi and Shravati AI, but we need to do more. I urge every scientist to engage with emerging technologies, not necessarily to master them, but to understand the limits to which we can push, the questions that we ask. What do these new technologies allow us to ask that we couldn't ask 5 or 10 years ago? If we understand that, we can partner with organizations that have the necessary expertise or technical skills and develop things together. The third question is, who will our partners be? Our natural partners for what we do are the pharmaceutical industry and clinical organizations, and of course academic institutes, but we are very good at forging those collaborations. The Indian pharmaceutical industry meets nearly 20% of the world's needs today. Dr. Reddy's laboratories and other companies have taken India to the world and built a solid reputation for quality and affordability. However, a deeper study reveals a dependence on imports for key starting materials and intermediates. Further, when we look at India's contribution to new drug development, it continues to be weak. It is here that the role of Indian academic institutions like CDRI comes to the fore. The Indian pharma industry needs to view academic institutions as their partners in innovation, where cutting-edge ideas can be tested and those with proof of concept taken forward by industry. For this to happen seamlessly, we need commitment, openness to experiment, and a common vision for what India and the world need. What I mean is that industry and academia need to collaborate from the start and agree on problem statements, target product profiles, and research strategies. Further, there must be a clear understanding of the funding model and revenue sharing. I can say that we have taken baby steps in this direction, both with Zydus and with Dr. Reddy's. We hope to expand this network of alliances to drive a new era of product development. In addition to the pharma industry, we must work closely with clinicians, and I think that was very apparent in the oration today. Our current list of clinical collaborators is quite substantial. They belong to institutes such as KGMU and SGPGI in Lucknow, AIIMS New Delhi, Manipal Academy of Higher Education, within that the Kasturba Medical College and Hospital, Tata Memorial, KEM Mumbai, PGI Chandigarh, NIMS Hyderabad, and several others. We must seek inputs and engage in continuous dialogue with clinicians to understand therapy and diagnostic gaps. To all our scientists, I would say, let us each find a clinical partner or two to stay in step with emerging unmet needs. Finally, let us remember that as India moves forward with the objective of becoming a developed nation by 2047, biomedical research will play an essential role, and we must contribute. Our research cannot count as scientific milestones alone. It must deliver solutions that address India's public health needs. Towards this end, let us move forward with purpose, conviction, and clear goals. Thank you.
Thank you, ma'am, for highlighting the important milestones in the last year and also sharing your reflections about how we can have greater value addition in the current ecosystem. With this, now we move on to the much awaited talk by Mr. G.V. Prasad. G.V. Prasad is a co-chairman and managing director of Dr. Reddy's laboratory, and you would be surprised to know that he is a chemical engineer by training and also holds a degree in industrial administration from Purdue University. He has covered a long journey making transformational changes in the ecosystem. He joined Dr. Reddy's laboratory in 1990 and then led the company from a mid-sized domestic operation to a diversified, trusted global pharma with presence in 66 countries with a total revenue of $2.83 billion. Mr. Prasad's emphasis has always been on research, innovation, transparency, business ethics, and he's a strong advocate of progressive people practices and high standards of corporate governance. As a managing director, Mr. Prasad is actively involved in hand-holding as well as mentoring of younger generation for delivering sustainable healthcare in the Indian system. He's a photography enthusiast and supports wildlife. He's board of members of Indian School of Business and Ashoka University. And he has been featured and his journey has been covered in many business magazines. And today we look forward to hear from your experience and your journey. Over to you, Mr. Prasad.
It's fine. I don't need a pointer. Thank you very much for that warm welcome. I also want to thank CDRI for hosting me today. I'm very, very impressed with everything that's happening here, and I'm really cursing myself why I didn't visit earlier. It's very gratifying to see the amount of contributions you guys have made to the pharmaceutical industry, for disease, understanding, and many other things. And I must say that I was very, very positively impressed and surprised that I didn't know so much about CDRI. So thank you. I also want to thank Mrugank and Divya for enabling my travel here, and of course to Radha for calling me here. You opened my eyes. Thank you so much. I'm also very proud of Radha. She's Dr. Reddy's alumnus, one of our brightest stars. And I'm so glad she's leading this institution. We can't take much credit for that, but we are very proud of you. She was always very bright, very smart, and very driven. So, CDRI, you are lucky to have her. My talk is not about science. I'm not a scientist. Though when I was a child, I used to tell my parents I want to be a scientist. I never got there. But I ended up in an industry where science is very, very important. And actually is a foundation of our business. So in that sense, I've been very engaged with the scientists, but I'm not a scientist. Coming in here, I didn't understand the power of what you guys have done. But I read up and I saw that you have your own complete infrastructure. You played a key role in developing many drugs which India needed. And of course, you have success stories. But I still feel the potential that I see in this place is much beyond what you've achieved. Radha said 10 ANDAs in the next 25 years, you said? That's a bit long. I think it should be more like 25 or 50 ANDAs. I think you can do it. You have the wherewithal, you have the science, you have the passion more than anything else. So I think, it's a modest target. I'm sure you'll exceed it many, many times. So Radha talked about India's pharmaceutical industry. It is one of the largest contributors of revenue and exports. But at the same time, if you take the global perspective, the value share is very low. The volume share is the third largest. But in terms of volume, in terms of value, we may be 15 or so. In terms of exports, I think the number here is 11. But if you actually quantify the full value of the pharmaceutical industry, or pharmaceuticals, we may be 15 or even less. This is because we operate largely in the generic industry, where the prices are low. Not only that, I think we had a great run in the last 20 years. We built the global generics industry from India. It's a globally competitive industry. It met a lot of needs for affordability across the world. But it's also a very intensely competitive industry because everybody knows the game now. They know process development, they know registration, bioequivalent studies. This is a very simple game, except maybe some difficult compounds like peptides or some special molecules. By and large, it has been commoditized. And we don't have to blame anybody, it's competition. That's how it works. The maturity of industry, commoditization happens, then only those who are, a few of them will emerge and succeed. And even those who succeed won't have great margins. If you see the pharmaceutical industry in the last decade, we had huge growth, we had huge margins. And sometimes, we very impressed with ourselves that profitability is so good in the industry. Not so anymore. The API industry is under huge pressure. The generic industry in the two big markets, Europe and US, is facing price pressure. People almost are operating at very, very thin margins. The saving grace is the emerging markets, in markets like India, which still grow and which still are branded, so you can retain that long tail. So the only solution to this is for companies like us is to innovate. And innovation is not easy. We are used to making a large number of small bets in terms of products. Each product development takes maybe $3 million, $4 million to develop, and then you file, wait for approval, then you launch. And you get, if you're very lucky, tens of millions or single-digit millions of dollars with good margins. But now we are looking at, if it's innovation, it's going to be a small number of very large bets. The fundamental risk profile of the industry is very, very different. And it's not like people like to say, it's not an evolution from generics to incremental innovation to discovery. It looks like a nice linear curve, but it is really not. It's a different game. And I think this is going to be a defining challenge for Indian companies who can make this transition from the commoditized generics business into a value-adding innovation business. Innovation can be of many kinds. Biobetters, biosimilars are also innovative because the skills are still developing. Incremental innovation in terms of formulation, and also breakthrough innovation in terms of drugs which actually change the standard of care. And there are a lot of new capabilities that the industry needs to acquire, and this is where I think academic institutions like CDRI can help the industry make this transition. So at the same time, I think the industry is financially very stable, making good profits. The economic environment in the country has dramatically changed. There's a lot more funding, risk capital available. And in the pharmaceutical industry is an important part of this journey. And so we still haven't made a mark on drug discovery. A few companies have done a little bit, but overall, it's wide open, especially when you compare to successful countries like China, who have made this transition so fast and so rapidly, and today have become a global leading force. After the US, China is the biggest innovator in pharmaceuticals today. So in what I just described, I think it's very important, not just from a financial perspective or a successful company's perspective, but really to other social needs. We need to address the disease burden, which is unique to India. Not many companies in the world are working on diseases which are very highly prevalent in India, infectious diseases, cancers which are specific to India, oral cancers, things like that. And then there's also, you mentioned about import dependency. That is something certainly there. And how many people can afford fully priced patented drugs in this country is a big question we all need to ask ourselves. And of course, if you innovate, you create value, it also creates prosperity for the nation and greater employment. And then there is the issue of access. So all these are not just financial goals, but actually social goals for the country. And with the emerging capabilities, with all the capabilities that have to be built as we served innovators through services, through CDMO, CRO services, and our academic institutions and our research institutions, we are well positioned to do this, and the time is ripe now. Oops. I think my slide is stuck. Okay. I'm going to skip this. I already covered it. There are challenges, though. The cost of, like we talked about, the bet on each drug is $1 billion or even more. And of course, there's a big risk. And right now, if you look at a phase 3 cost, and if you do it at a global level, it's hundreds of millions of dollars. And there are gaps. We don't have the ability to take such large risks. And if you see our biotech industry, we are still very, very small compared to what is out there in the world. And we are not at the rapidly scaling that up. Of course, there are some centers of innovation in India which are doing better than others. But overall, the startup scene is still not as vibrant as the IT companies, IT sector startups are. There are, of course, regulatory challenges. Though the drug controller is one of our ex-colleagues, Radha knows him too, he was also an alum. He's trying to do his best, but still it takes a lot of time to get approvals. And anything they don't have a precedent. It takes an enormous amount of debate, challenge, rejection, reapplication, all of that. They're not really adapting to the changing regulatory environment, and that is something we need to do, because the staffing of CDRI, of DCGI is largely non-technical people, and for them to learn, understand, and they also use external help, but they're also largely academics who have never done drug discovery in their life. So this is something that has to change and become a more enabling environment. You talked about hospitals, yeah. So while there is collaboration between some hospitals and research institutions and pharmaceutical industry, it is not at the level at which we need it. The whole corporate hospital system, they have very little time for research. And they have a bulk of the patients, and doctors doing research along with pharmaceutical companies would be a big value add for India. And that's something the few hospitals are doing, but not the majority of them. And finally, I think as a government institution, as a public academic institution, there are so many restrictions. And I don't know, you didn't speak about restrictions, but looking in from outside, there are a lot of constraints of decision making, of complying with government rules, and incentives are limited. Financial freedom is limited. With all this, you're doing so much. I must applaud you. So it's difficult to do this. It's very difficult to do this. And I hope industry and academia work together well to solve some of these things and make up for the constraints of working in a governmental setup. And some of the things I talk about here, I already spoke about, enhanced industry-academia collaboration. I myself am surprised that we didn't reach out earlier to CDRI. There's a lot we can do together. Maybe, industry has to also visit the institutions, understand the capabilities, and not have a generalized view of academic institutions. So maybe industry has to do this, and also you people have to sell yourselves more stronger and harder, and probably call us here, bring us here, so that we can see what's happening. You talked about AI. We are actually starting to use AI and seeing remarkable difference in capabilities, at least for the starting points, for using AI-driven drug design, AI-driven process development, modeling and simulation, computer vision. All of this are coming together and making drug discovery much better, much more productive. Preclinical models, I'm not going to talk about it. It's not my area, but I think there are advanced models. We saw the organoids stuff that Sandhya spoke about. Financing is another thing. Companies need financing. Academic institutions need much more financing than you have. And also make your collaborations global. Sandhya, you spoke about your collaborator who was in Europe. And I think institutions like yours also can build these kind of connections. The last point is very important. And this is the reason China succeeded, that they developed a very attractive domestic market for innovative products. They put them on the reimbursement list. They gave them high pricing. They gave them protection. Depending on the level of innovation, different levels of protection. And that created a lot of financial incentive for companies to really invest in. So they first did the innovation for their country, then now they're going global. And this whole transformation happened in 10, 15 years. And if India does this, I think we are all equipped to do an equal or even a better job than China. Today, if you look at what China's done, I think within maybe 15 years at most, they've become the second largest innovator in the world. But we do have some success stories, and if we build on these success stories and take products from the lab to a global launch, it'll create a positive spiral. Dr. Reddy's was one of the early movers in drug discovery. It was a magical place, Dr. Reddy's Foundation, when the team was working there. We started with analog research, but we graduated into other areas also. Zydus saroglitazole is now launched in India. It's being launched in the US. So there is a very great deal of excitement around that, and a few other products here. I want to take the opportunity to plug Dr. Reddy's a little bit and what we did, how we emerged, and then how we made mistakes and what we learned from them. Initially, people who know my father-in-law, the founder of Dr. Reddy's Labs, he was a scientist, he was a chemical technologist, but he took great interest in drug discovery. He was very passionate about discovering new drugs, and he started this effort in the mid-90s, 93, 94. He started in 95, we were operational, with full infrastructure, including animal house, med chem, analytical chem, and biology labs, everything. Focused on analog-based research. I think his first effort was trying to do a better fluoroquinolone, which didn't succeed, but then went into diabetes, worked on the class of compounds known as thiazolidinediones. Quite successful, actually licensed out products to Novo and Novartis. Unfortunately, in the phase 3, one of them failed. And it's kind of, after that, we kind of drifted into cardiovascular, difficult areas of research. And we couldn't make a big difference there, and we had to wind down that operation. Radha will remember I was one of the guys who was involved in shutting that down and moving on. But that's something we had to do because we were not seeing the path to market from the research. And also, I think early on we were a bit naive to think we could do everything internally, do the clinical development, and then take products to market. Also, we were looking, another mistake we used to make was go from the science forward rather than the patient backward or the disease backward. We didn't have a target product profile or anything, but we were just learning, doing and iterating, and it was quite an expensive effort. After that, we tried to get back into innovation through incremental innovation. So we focused on the US market, understood the liabilities that an asset had or a product had, and tried to work and improve those liabilities through innovation, either through things like reducing the dose sparing thing, or from an injectable to an oral, or instead of 3 times a day, once a day, those kind of things we looked at. Fairly successful effort. We had actually 6 NDAs approved in the US, new drug applications approved in the US. We were also starting to market, but then the company went into a fiscal problem with some issues with the FDA. We had inspections, bad inspections, warning letter, and all of that, and then we had to pause that, and we actually had to sell that business off because we were needing to fund a lot of it, and the company didn't have that wherewithal at that time. And along the way, we also started Origin. Some of you know Origin here. We started as a structure-guided drug design company, trying to solve proteins whose structures were not determined at that time, and try to do drug design around that. Over time, we saw that was also not such a hot area for customers, and we started building services that the customers wanted. And we had a set of services in medicinal chemistry as well as drug discovery, full drug discovery collaboration, and also licensing out as a model. And finally, in the last 7-8 years, we stopped licensing out and started doing clinical development, and I'll talk about it in a little while. Of course, the services business was spun off as Origin Pharmaceutical Services Limited. We also saw that we can't innovate a lot, so we actually thought that it's a good idea to partner with other innovators and bring their assets into markets which were underserved by them. Markets like India, Africa, Middle East, South America, which were not the large focus for the innovators, and this were mostly distracting them, so they were open to license those products. And we do a lot of licensing with China these days. The Chinese companies are much more open to collaboration with Indian companies than large Western multinationals. So we built a nice portfolio of innovative assets which others innovated, but we brought them to market. So I want to take a little bit to talk about Origin Oncology. We work on only one therapeutic area, which is oncology, and that too within that immune oncology. We have about 400 scientists. Our pipeline has about 10 or 11 clinical assets, and the rest are preclinical, about 20 totally. There are various platforms here. I'm not going to go into that, but we are focused on cancer, but all modalities: small molecules, large molecules, MABs, ADCs, bispecifics. We are also doing a CAR-T there. So we focus on one disease, but are covering all the modalities there. We worked as we learned the business from moving from services to discovery. We worked with a large number of very important companies in the world, and in that process gained experience, expertise, and the processes itself, and that has helped us immensely as we built our own pipeline. We also had some licensing deals with early stage licensing, which are described at the bottom of the slide. So I'm not going to go through this except to highlight that we have one asset which is in the final stages of the phase 3. In a matter of few weeks, we should know the results. We are waiting with bated breath, and we hope it will be successful. This is an India-only trial. If it's successful, we'll do a global trial and launch this product worldwide. That is our plan, to transform to become a oncology discovery and marketing and commercial global company. This is our preclinical pipeline here. We've stopped out-licensing unless it is an asset which we can't develop at all because of the nature of the product and the kind of trial it requires. But otherwise, we are doing up to phase 2 at least, establishing the POC, because the value inflection really takes place when the asset has passed the POC in humans. If you do it early, you get less money. If you do after phase 1 also, you get a little more money, but not more. The big inflection takes when you have human POC. And we are thinking of that as the strategy as we go forward. That is about Origin. I think as a country, we can learn from other countries. Of course, US is the leader in innovation, and its wonderful university system, public funding, NIH funding, all this have helped build a very strong and vibrant industry. But there are other innovators we can learn from who have done it in a different ways. China, as I said, has done a lot, and we can learn from them. Of course, another country which I recently visited just for study their innovation system was Israel, and was very impressed with how some little money made such a big difference to them. I'm going to skip these slides, a lot of detail here, but broadly, I think government policy on talent, on funding, on regulatory, and creating a local market. These are the lessons we can learn from China. Israel is a little different. They're largely a startup nation with incubators within hospitals. And within their academic institution, like Weizmann has its own incubator. And the private equity guy sits in Weizmann. So there's VC, private equity, academic research, and the hospital system combining together to create, a very vibrant culture of startups and innovative companies. And this is also something we could do. I can see, green shoots in Bangalore especially. There's a lot of biotech activity coming up, and I'm sure some of these will be very successful and scale up. So we need all of us to work together. We need industry to reach out to academia to work with us, and government to help us cross all these barriers. As industry, I think we must commit ourselves to innovation. We are still, dipping our toe, but not really fully getting into it. We have to partner, not just in India, but across the world. We must look at innovation as the next wave of growth for the industry. And of course, we have to manage that risk. We could start doing many things with academics and reduce the risk before we really jump into it. Academia, like I said, I think you may want to specialize a bit. And I was looking at your budgets and the number of TAs you're in. It's a lot of work. I mean, a lot of diversity of therapeutic areas. Unless you get a lot more money, I'm not sure how that will be scaled. Of course, horizontal collaboration is something which will be valuable. And again, is there a way we can incentivize innovation and entrepreneurship? Entrepreneurship is very important for this industry. You need somebody who is focused on the value creation, who can take the resources, combine them in ways creatively to create nonlinear value and see the end outcome. And maybe an entrepreneurship cell within academic institutions will help. Of course, government, I talked about regulatory frameworks. I think they also, instead of just throwing money at innovation, they could create a system where successful companies make a lot of money. Successful investors, individuals, and companies make a lot more money, and to get them there, funding them there through milestone-driven payments, whatever. But that's something I think if the government does, it will build the industry much more rapidly. So I think to my peers in the industry, we have to look outward, we have to collaborate, we have to invest, and we have to also persist and not give up because of failures. To the governments, I think they have to modernize their regulatory frameworks. They have to enhance, create enabling conditions for industry and academia partnerships, and see how AI can also be powered up so that India can use those tools to really move innovation forward. And I think as CDRI, you already are doing a lot. And I think you can see how you can market better, understand the end outcome, work backwards, and see how you can connect the dots to create value. And we can, like you said about 2047, the big goal of making India a power in innovation and discovery research, it's very much possible. I wanted to end with this quote. As I mentioned, Dr. Reddy was a strong proponent of drug discovery, and he almost called it a spiritual pursuit. And I think you need that kind of passion to innovate, because this work is very difficult, very complicated, very long term, and we need that kind of passionate entrepreneurs, scientists working together to make this possible. We still don't have a product which we developed all the way from lab to global launch, and that I think will be the defining moment for Indian industry. And with that, I want to end. Thank you very much for giving me this opportunity. Shall I take it out?
Yes.
This is mine? Or
No,
this is yours.
So please remain on the stage.
Okay.
Thank you, sir, for highlighting the key challenges as well as the opportunities for the Indian drug discovery ecosystem. If I may ask a question to you, we heard Dr. Sandhya Vishweshwarya saying, 25 years later, they see that, whatever was done had so much of value addition. How do you deal with the problems like, sometimes you have really good projects which are close to your heart, but then you have to close it down and you have to tell your team, this. So how do you deal with these situations?
I learned a lesson from Radha today. She said she has a committee which does that. But I think it is a difficult thing to do. It is, but that is what the role of leadership is, to see when we've reached the end of the road and take a different direction.
And so still now, India is still considered, a generic manufacturing country. We have not been able to get into the NCEs.
Yeah.
What are your thoughts about it, and when would the Indian farmer have that,
Yeah, so
the
I
step
don't
forward?
I don't want to underplay the accomplishments of the industry. Generic industry is not an easy industry.
Yes.
Yes, it has a different kind of difficulties. You need to have cost leadership. You need to be first to market. You need to have a portfolio consisting of hundreds of products, and then you have to market it around the world, the logistical challenges of that. So we should be proud of what we've accomplished in the generic industry. India is a big force in generics. Of course, it's not where the nonlinear growth will happen. The nonlinear growth will happen when you have innovative products. And I'm not sure it's just a road that goes up from APIs to innovative, to generics, to biosimilars, to innovative products. I think it's a different game. Many companies are discovering that. So they're using financial resources to buy portfolios of products, licensing in. A number of different parts of the puzzle are being assembled to build a new business. We are also struggling with that question of how to build an innovative. We are now tasting a little bit of success through licensing in. We are learning from the emerging markets. We are also taking on some products for the first world markets, like we have a product which we want to launch in Australia, which is like a reimbursed first world market, and we are gaining that experience. And at some point, I think we will be ready, have the financial wherewithal to take a product from the lab to a global launch. And I think that should happen in the next 10 years, within the next 10 years.
And we would also be happy to be part of that journey. Our young talent sitting in the audience
Yes,
would
absolutely,
be contributing to
Yeah.
that. Okay, so once again, let's give a big applause to
Thank you.
Mr. Prasad. I would now call upon Director Dr. Radha Rangarajan to felicitate the speaker. I request Dr. Vishweshwarya to also join on the stage, and Divya to please come on the stage. I request the dignitaries to please remain on the stage, and we will now move on to the next section, and Muktada would be taking the program forward for MOU and tech transfers.
Thank you so much, ma'am. So, good afternoon, everyone. CDRI is signing a memorandum of understanding with the Center for High Impact Neuroscience and Translational Applications, Chinta, Kolkata, for joint research in neurosciences. Therefore, I invite Director Chinta, Dr. Sumant Sona Chatterjee, Director CDRI, Dr. Radha Rangarajan, Dr. P.N. Yadav, Head of Neurosciences, and Dr. Naseem Ahmed Siddiqui, Head of Business Development for exchange of MOUs. This collaboration aims to develop Alzheimer's disease-specific brain organoids for research and therapy. Thank you so much. Moving on, we have the technology transfer segment. As told by Director Ma'am during her address, CDRI has successfully licensed the technology for an indigenous RT-PCR kit for simultaneous detection of arboviruses, dengue, chikungunya, and Zika to MyLab Discovery Solutions, Pune. This licensing took place at CSIR Innovation Complex in Mumbai on January 17, 2025, in the august presence of DG CSIR and other dignitaries. Today, we are transferring the technology to MyLab Discovery Solutions, Pune. The demonstration of the technology was held during 14 to 15 February in CDRI. May I now request the representatives of MyLab, Dr. Makhan Kumar, AGM, and Ms. Amruta Joglekar, the Quality Control Officer, to please come up on stage. Also, may I have the pleasure to invite our very own CDRI team, led by Director Dr. Radha Rangarajan, Dr. Naseem Ahmed Siddiqui, Head of Business Development, Dr. Atul Goyal, Dr. Ashish Arora, Dr. Neeti Kumar, the investigators of the technology and their students, Priyanka Pandey, Seema Vidyarthi, Nirupa Chaurasia, Ramya Pandey, Vikas Yadav, and Aradhana Chauhan to join us all on stage. Can we please have a great round of applause for the entire team? Thank you so much. And now I request the director and the chief guest and the dignitaries to please remain on stage for the felicitation of our superannuated staff. Ankita and Megha will take it forward from here. Can we please have the AV of the technology transfer that took place? The video, please.
Now we are going to felicitate the CDRI employees who have completed their valuable 25 years of service in this institution. For that, I would like to request our orator, Professor Sandhya Vishwashwarya, to do the honor. Mr. Neelambhur Shankar Prasad, Store and Purchase Officer. Mr. Vinay Kumar, Controller, Store and Purchase. Mr. Ramesh Chandra Gupta, Senior Technical Officer, Knowledge Resource Center. Moving on, now I would like to invite our colleague and CDRI family members who are superannuated last year. So I like to request our chief guest, Mr. G.V. Prasad, to do the honor. Dr. Sharad Sharma, Chief Scientist, Division of Toxicology and Experimental Medicine. Mr. D.K. Tripathi, Senior Technician, MMI Division. Mr. M.P.S. Negi, Senior Technician, Division of Toxicology and Experimental Medicine. Mrs. Sufia Kirmani, Section Officer, E1. Mr. Mahesh Chandra Verma, Assistant Section Officer, General Section. Mrs. Rani, Assistant Section Officer, General Section. Mr. Ravi Kumar Mehra, Senior Technician, Store and Purchase. Mr. Yashwant Kumar Singh, CDRI Canteen. Mrs. Anupama, Senior Technician, Division of Toxicology and Experimental Medicine. Mr. J.K. Joshi. Mrs. Deepali Pandey, Principal Technical Officer, MPC. Mr. Gangaram Yadav, CDRI Canteen. Next, I'd like to invite Mr. J.C. Rajan, Lab Assistant, Medicinal and Process Chemistry Division. Mr. Ravi Shankar Mishra, Molecular Microbiology and Immunology Division. Mr. S.C. Tiwari, Senior Technician, MPC Division. Mr. Mohammad Islam, CDRI Guest House. Mr. Najibullah, Animal House Facility. Mr. Dilip Kumar Sen, Assistant Section Officer E2. Mr. Dinesh Pal Singh, CDRI Canteen. Mr. Ramesh Chandra, Senior Technician, Division of Pharmacology. Mr. Gaya Prasad, Laboratory Engineering Services.
Mrs. Kanchan Bala, Senior Secretariat Assistant, Store and Purchase. Mr. Rajkumar, CDRI Canteen. Mr. Harish Kumar, Senior Technician, ESD Division. Mr. Ramesh Chandra, Vigilance Division. Mrs. Seema Rani Srivastav, General Section. Dr. Harsh Mohan Gunyal, Principal Technical Officer, SAF Division. Mrs. Renuka Mushran, Neuroscience and Aging Biology. Mr. Anup Kishore Pandey, Senior Technician, MPC Division. Thank you to all the superannuated CDRI family members. And thank you to all the dignitaries. Now I'd like to request you to all to take the seat. And now may I request Dr. Divya Singh to deliver the vote of thanks. Thank you.
Good afternoon, everyone. I am pleased to present my vote of thanks on the occasion of CDRI's 74th Annual Day function. On behalf of the Annual Day Organizing Committee, I begin by conveying my heartfelt gratitude to our Director, Dr. Radha Rangarajan, for her guidance, unwavering support, and encouragement for the successful organization of this event. Ma'am, you have been very generous with your time whenever needed, regardless of your occupancy. So thank you very much for all the inspiration and support without which this event was not possible. My special thanks also to Dr. Sandhya Visvesvaraya for delivering the 50th Sir Edward Mellanby oration, and Mr. G.V. Prasad for being the chief guest of our annual day function. Thank you to both of you for generously accepting our invitation. It was a wonderful opportunity for us to hear your thoughts, and I'm sure this will encourage all the young minds, all the students to traverse the path of fundamental science and drug discovery. It has been an honor and a pleasure to have both of you. Nothing can be completed without the dynamic support and backing of a proactive and devoted organizing team members of our institute. I have been fortunate to have the support of the team that was involved in managing the annual day function, and I want to thank each of them. I would begin by thanking my co-convener, Dr. Mrigank Srivastav. His unrelenting support and constant motivation have been a great encouragement, and this helped in the successful organization of this event, and without his support, this event would not have been possible. Thank you so much, Mrigank. I also I wish to thank all the conveners of the subcommittee, which include Dr. S.K. Rath, Dr. Vinita Tripathi, Dr. Deepak Datta, Dr. Anand Kulkarni, Dr. Richa Pandey, Dr. Anil Gaikwad, Dr. Kishore Mohanan, and Dr. Prabhat Mishra. Especially, I would like to thank Dr. Rajkamal Tripathi. Whenever there were any hurdles or any ups and downs, he was always there. He always supported us. So a big thanks to Dr. Rajkamal Tripathi. I also want to express my sincere gratitude to COA, COFA, COSP, and SOG, entire PME division, especially Mr. Ravindranath and Mr. Kazim for their help and support, staff of the director's office, staff from the admin, finance and purchase section who helped with their guidance and support whenever it was needed. I also gratefully acknowledge Dr. Neeti Kumar's help and time for anchoring today's event, and also the staff of the audio and visual section, our photography team, and the CDRI canteen and guest house for their help and support. I would also like to convey my gratitude to the media personnel, and I would be failing in my duty if I do not thank all the student volunteers who have contributed this annual day a grand success. And finally, I thank everyone here for taking their time to join us today. I think it has been a wonderful audience. A big thanks to everyone. And I would like to say that there were times when the things were tough, but I think we managed it well. And it is said that when the going gets tough, the tough gets going. So as we conclude this event, I request everyone to stand up for the national anthem. And also, I would request that after the national anthem, please stay back for a group photograph. Thank you.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Kazim, make group photograph.